All right, so these are the four phases that we just mentioned, including phase 0. So it's really 5 steps or so. And I really just want to point out here on this slide the duration of a contractile cell action potential. It's much longer than skeletal muscle, right? And we mentioned that earlier. These are much more slow, sustained contractions for smooth and cardiac muscle than what we saw with skeletal muscle. So it's much, much longer than the duration of skeletal muscle. And that is really for allowing calcium to get into the cell, okay? All right, so this graph helps to illustrate what we just drew. So we start at rest here, rapidly depolarize in phase 0. We start a brief repolarizing period, and then we interrupt that with the plateau phase, and then we continue on repolarizing right back down to rest. Okay, so 2 important distinctions here is that sodium is responsible for the depolarizing here, okay? There's a plateau phase that allows for calcium, and then also understanding that we start a little bit more negative in a contractile cell than we do in a pacemaker cell, okay? All right, so what we're gonna move on to speak about now is really putting that together, looking at excitation-contraction coupling, and that is basically very similar to what it meant in skeletal muscle. What happens in between the time that the action potential gets to this cardiac muscle cell and we actually see contraction? So we have a lot of the same structures here that we had in skeletal muscle, the T-tubules, the SR, which is only part of the source of calcium. We also have troponin and tropomyosin interaction, which regulates this process, right, turns it on and off. Then we have gap junctions, right? These are properties that are similar with smooth muscle, the presence of gap junctions and the presence of extracellular calcium, all right? I kind of mentioned before to draw a Venn diagram as a study tool to kind of see what properties skeletal muscle and cardiac muscle share, what properties cardiac muscle and smooth muscle share, and then what properties smooth muscle and skeletal muscle share, right? Because they all share a few properties. All right, so let's walk through the actual process of excitation coupling here. So first thing we want to point out is that we start with the impulse, the action potential, either on an autorhythmic cell, which is a pacemaker cell, or on another contractile cell. All right, so a pacemaker cell is generating the rhythm, but we can also have a contractile cell that is propagating that rhythm as well. And so the current passes through this gap junction that we mentioned and arrives here on our contractile cell. Okay, so we only see this machinery that we're going to talk about on our contractile cells. Now, when the current gets here, the action potential that is, we stimulate the opening of these calcium channels here. Okay, we talked about that, right? We just looked at that, and that allows time for calcium to get into the cell. When calcium comes in, it's coming from 2 places, right? It's coming from the SR, but also most of it coming from the extracellular fluid. And that is going to interact with troponin. Okay, don't forget that stuff we talked about. Interact with troponin, drag tropomyosin out of the way. That is going to elicit cross-bridge cycling. We already know this process in detail. And then we're going to cease this contraction by getting rid of that calcium, okay? So we have 2 mechanisms for getting rid of the calcium. We can pump it back into the SR, right, actively using a calcium ATPase pump, or we can exchange it or pump it out of the plasma membrane, okay? So we can either exchange it for calcium here, okay, the secondary active transport, or we can simply pump it out of the membrane itself using that calcium ATPase pump. Right? But just important to understand, we must, just like we saw with skeletal muscle and smooth muscle, we must get rid of the calcium in the cell in order to cease muscle contraction. Okay, so that's pretty much what this slide is reiterating. We must remove the calcium from the cytosol. We can do it via 3 mechanisms. We can pump it into the SR, we can pump it back across the membrane, or we can exchange it for sodium at the membrane. And so this whole thing happens, we already talked about this, troponin and tropomyosin, and how they regulate this process very heavily.